Hello, Sujit sir, are you there?
Here. Yep.
Sir, your voice is audible. Can you share your screen?
Yeah. Just a minute.
Yeah.
Hello.
Yeah, Sujit sir. Sir, are you okay with the screen share?
Just a minute. Just a minute. I'm just connecting with
Okay,
my
no problem.
headset.
ఫస్ట్ స్లైడ్స్ షేర్ చేసి పెట్టుకోండి. ఒక 2 మినిట్స్ తర్వాత అంటే 1 మినిట్ ఇట్లా ఫోటో తీసుకున్న తర్వాత
I hope my screen is visible, madam. Hello.
sir.
My screen is visible,
Yes, sir. It's perfect.
and my voice is also audible to everyone.
Everything is perfect, sir.
Thanks.
Sir, can you stop your screen share for a while? Hiranma? Sujit sir, are you getting my voice?
Yes, I'm getting your voice.
Can you stop your screen share for a while? If you don't mind.
Stop it. Okay, okay.
Yeah.
Yep.
Vishnu sir, are you hearing me?
Yes, madam.
Can
Dr.
you
Sujit
uh,
Garu, welcome to our session.
can you on your video and start the presentation section?
Sai Vishnu, please start your session.
Okay, sir.
Sir, video on చేయండి.
Okay, ma'am.
Yes, sir.
Good afternoon to all. Myself, M. Sai Vishnu. I am here to introduce the Dr. Sujit Tangarpallivar. Dr. Sujit Tangarpallivar is a founder of Pharma Toppers and co-founder of IT company Octane Private Limited. Currently, he is working as a data scientist on computer vision and natural language processing projects at Extensia, based out of Oxford, United Kingdom. He served in multiple positions in many companies before, like Persistent Systems, Accelera, Hexaware, React Matrix, and he played a crucial role in applying artificial intelligence in the drug discovery field. He obtained his PhD in pharmacoinformatics from National Institute of Pharmaceutical Education and Research, Mohali. He published several research papers in peer-reviewed journals and developed industry-usable softwares in the field of chemoinformatics and bioinformatics in his industry and academic tenure. He has been using his programming skills in solving biological and chemical problems for a long time. Now, I hand over the session to the speaker, Dr. Sujit Tangarpalliwal.
Thank you very much, Vishnu sir, and for introducing me with a very nice words. And good afternoon, everyone, all of you over here. So my voice is audible to everyone, right?
Dr. Sujit sir, you were audible.
Perfect. Thank you. Thank you. Thanks for confirming. Okay. So as we discuss as the top initially, I would really would like to thank each and everyone who was as an organizing committee for this conference, and also would like to thank for inviting me for this such an national level conference to deliver my work, what I have done, so far in the world in the field of drug discovery and artificial intelligence. Thank you, everyone. So to start with the session, let me share my screen. I hope my screen is also visible to everyone.
Okay, sir.
Yes,
Yep.
sir, it's perfect.
Sorry.
It is perfect, sir. No.
Thank you. Thank you very much. So before starting a session, I just wanted to have a small survey on how many participants we have from industry, how many participants we have from academia.
Sir, all the participants are from academics itself.
Okay, all the participants are from the academia. That's fine. And I just wanted to know how many participants we have who knows artificial intelligence, who knows data science, who knows programming, and or how many participants we have who knows the biology, chemistry, and artificial or data science, all three together. Because if you could see in my screen, we have a three different fields: chemistry, biology, and artificial intelligence, which also called as a data science. So how many participants we have who knows all this knowledge of all these three? Those who are attending this session, please unmute yourself and you can speak out if you have any problem during my entire session. You can interrupt me at any point of time, and also please speak out if you or you can type in a chat box that you if you know about artificial intelligence one, if you know about biology two, if you know about chemistry three. If you know all these three, you just type 1 2 3. If you know any one of this, you type one number, two number, or three number, please. Just so that I'll take you to that depth level, if based on your understanding and based on your knowledge. You can just type one or two or three or one two or one three or two three or 1 2 3. Please quickly, guys. That's perfect. Fine, fine. I could see many people are from the biology and from the chemistry area, and very few from the data science area who knows about data science. So I expect you guys knows a little bit of a programming. Uh, so to start with, to work on a data science, to work on artificial intelligence, you should know programming. Along with that, you should know mathematics, and you should know a lot about chemistry and biology, definitely, if you are working on AI in pharmacy or AI in biology or chemistry. So so for today's session, I don't want to present some definitions, some common terminologies in artificial intelligence or biology or chemistry. I would directly jump into application of artificial intelligence, how Indian pharma companies or uh, international pharma companies or other life science companies, they are using artificial intelligence or data science in their day-to-day activity. So I'll discuss more about what I have done so far in this industry, in the how we have expedite the process of drug discovery using AI. I hope each and every one of you know a drug discovery is a very long process. It takes around 15 years, 20 years time from the target identification or from the very first step to the up to the last step where company gets an approval from the FDA and drug gets launched into the market, right? And it costs billions of dollars to find out one drug and to get launched into the market. So this entire process and because it's cost consuming, that's why many pharmaceutical companies were they were not entering into drug discovery cycle or drug discovery stage, specifically I would say Indian company in the previous time. But because of the artificial intelligence, the time duration of a drug invention has got reduced. It has got to around 10 years or 12 years or even lesser. And the cost which is actually involved in drug discovery or development has again got reduced. So in previously it was taking billions of dollars, but now it has got reduced to some extent. So it's all because I would say artificial intelligence or AI. So what exactly is happening? AI will allow you to predict what human can predict, but not with that accuracy, but yes, with that scale. If I'm a person, if I'm reading 100 different articles in 1 year, AI can read those articles in several minutes and he can give us a conclusion of those 100 different articles. Accuracy, I cannot sure it based on how you develop your model, but yes, AI can do with some extent. So artificial intelligence, this term was there in 1950s, but because of the data availability, because of all the resources, computational power, this field has got very boom in the market nowadays, and there are many companies I would say in the UK and US, who specifically startup companies, those who are working on AI and they are providing their services to large pharma companies like Pfizer, Novartis, and all that. But yes, along with that, these companies like Pfizer, Novartis, they have their own data science team as well. But yes, they are tying up with other other small companies or biotech companies to accelerate their drug discovery cycle or to accelerate their discovery process. So the presentation which I am going to share today is the work which I have done so far in my experience, how we expedite the drug discovery process. I am not gonna just reveal the company names for which I have worked. I have worked for on these projects and how it has got. But yes, definitely I'll give you all the knowledge about what industry does, what exactly we have. I don't know whether you have heard or not, but yes, I was listening to these words like artificial intelligence can solve many problems, can be useful to expedite the process, reduce the cost, and all these things. So let's see how we help pharma company to to get better results using AI. So that's why I'm directly jumping into that example without going into any definition or any basics of any terminology I'm expecting. But definitely during my presentation, if you have any question, any point at any point of time, you please raise your hand or you unmute yourself and please do ask question. I'll be happy to solve your basic problems. And definitely during my presentation as well, I'll explain some basic terminology, what exactly it is. So, as you know, guys, toxicity prediction is a very big challenge because in previous times, even now also, several animals are getting sacrificed to conduct the toxicity studies. So, if you could see, over 100 million animals are used by the cosmetic and drug companies in the United States each year. It's a huge number, 100 million. So, what are the limitations for the animal testing? The first thing is, if you know about European Union and some other regulation, they have completely banned for the animal testing. I mean, what their regulation is, any drug or any study should be conducted in computational first, and once it is approved or once it is successful, then and only we can conduct those tests on an animal. You cannot directly go into animal studies once you have your molecule or once you have your samples. So that's why we have a lot of cultural limitations. Many people love animal and all those. Biological limitations, we may have uh, animal shortages in animal, their species and all those stuff. And the chemical is the major, I would say, hurdle to conduct all these trials, because chemicals are costly and availability is the major hurdle. And the biggest problem is the economic limitation to do all this animal test, all those animal studies. We companies has to pay, or companies has to invest lot of money in their studies. So to avoid all these limitations, now companies are directly not going for the animal studies. Initially, they are screening their molecules, they are doing their studies on a computational level, on a computational. I'm sure most of you know computational in silico words. So companies are doing in silico studies first, and once it is approved or successful, then they are going for animal studies. Many companies. So what kind of studies we can do? There are several endpoints, I would say, or several toxicity studies which company has to do for any drug molecule, or for any drug molecule before they go further. So like I have just named few, like skin sensitivity, corrosion, eye irritation, mutagenicity, genotoxicity, all such kind of toxicity studies we have to companies has to do for their molecule. And so to avoid animals, companies are looking into in silico approaches or in silico software tools. So what problem or what question we have solved? So the problem is, can data from completed short-term preclinical studies in a particular indication be used to predict the treatment effect for the future long-term studies trial in the same population? So as you know, we are not talking about the clinical studies. Here I'm talking about the preclinical studies. I hope you understood about preclinical and clinical studies. So in the preclinical studies as well, we have several phases. Maybe sometimes a company might have to test with a very small number of animals or a small number of samples, and then they have to do another phase of trials with large population, large samples. So again, to do small studies or a large studies, company has to invest a lot of money. So we thought, why don't we predict? Let's say there are 3 or 4 phases of a trials, preclinical trials, a company wanted to do. And let's say first phase could be for based on the 10 or 15 animals, and second study would be for 100 animals, and third for maybe thousands of animals. So we came up with an approach. Why can't we predict second phase from the data of a first phase using an artificial intelligence? So this question is relevant to the situation where a go-no-go a decision to start a required long-term study trial being considered, but available phase 2 data is limited to short-term treatment. So in our study, let's say in our phase 1, if we got, okay, this particular molecule may cause toxicity, is causing a toxicity, so we can directly interpret for the second phase, yes, this molecule may cause toxicity to higher number of animals, or it may have those other kind of side effects or something. Because a drug may or may not show all its effects in a phase 1 study, but it can show, because if you know, some drugs may pass phase 1 clinical trials, but they may fail in a phase 2 or phase 3 clinical trials. Because if we test on a large number of population, we actually gets the data for everything. So that's why, but conducting those trials with a large number of population is a very uh, cost driven, so, and time driven as well. So to reduce that, we came up with the AI approach. So i'm I'm really happy to say that this is one of the very first kind of approach somebody has tried, as per our knowledge. When we proposed this solution to various companies, they said, we saw such kind of things very first time. So yes. And let me go to the next slide. So here we have used multimodality approach. You must have heard, many of you might have heard about uh, people, they take chemical data like drug molecules information, they calculate descriptors, and then they do some sort of normalization step, and they do training and test set splitting, and then they build a model with a machine learning algorithm. This is a very common practice which is happening in the field of chemoinformatics. So so everyone can do these things. Also, like, we can incorporate biological data as well, like protein sequence information or protein related information into that. You must be aware that we can calculate the fingerprints or a molecular features for the protein as well. So we can incorporate both this data or both this information here.
If you want to share that, so that's enough, sir, because I need to maintain the database.
Anybody's talking here? Do you have any questions?
Thank you so much.
Okay, fine. Yeah. So hope my screen is visible. Again, my presentation is visible.
Yes, sir.
Perfect.
You are
Thanks.
audible.
Perfect. Thanks. Yeah. So what we have done is, using a multimodality approach, we did not stick into only chemical space or a chemical data, which is called drug molecules. We also look it into biological data. We considered few proteins information, and genes information as well. And along with that, the another modality which we have is, biochemical data, imaging data, doses information, and in vivo data from the 1 to 3 months study clinical trials. Sorry, preclinical trials. So we have developed a lot of machine learning, deep learning models, and how NLP help us to get this data is. Uh, I hope you must know about NLP. NLP is a natural language processing. Uh, in a simple words, I would describe NLP as a NLP can understand your text information. NLP can understand your voice. NLP can understand what you are talking about. So, in a simple words, I can define NLP for you. But let's say if I am a programmer, if I am a AI scientist, so NLP, how I use NLP to get all this information. So using NLP, we retrieved all the chemical information from the publications without reading those publications manually. Because let's say for the skin toxicity, we got millions of publications from the PubMed and from PMC and all other sources. So we cannot go through each and every publication and read and extract, copy and paste those information from the research papers to our Excel sheet or our database. So in that case, NLP helped us a lot. NLP can detect in which line the molecule information is given. I mean, which line actually represents, or which sentence actually represents about the molecule and what kind of molecule it is. So we wrote some algorithm. So we got a lot of data, from chemical data, from the literature, and we analyzed a lot of papers as well. Using we built several deep learning models with NLP. Like we did text classification. We initially identified which articles is actually talking about the skin sensitization, which article is not. And then we did sentence prioritization or a sentence identification, which sent in the articles which sentence is actually talking about the skin sensitization, and we extracted the relevant information from those articles. The same for the biological data as well. Some proteins related to skin toxicity, or toxic proteins which are involved in the any of the mechanism of the skin toxicity or skin sensitization. So we got all this data from the natural language processing. So I intentionally haven't described this process in the slide, just because this is all driven by mathematics, with all those algorithms. So that's why I have explained you in this way. But yes, you can please go and please look into what is natural language processing and how exactly works. To talk about, I can give you even more brief about the natural language processing. Uh, you yeah, Google can convert your any language, let's say English to Hindi, or English to German. Any language Google can convert. So I would say this is all because of the natural language processing. If you have a one sentence, and if you want to predict the another sentence, still natural language can help you to generate the new sentences as well, or to predict what exactly the another sentence defines based on your previous sentences. That also can, such kind of things, natural language processing, NLP, can do. Okay. So so this chemical data was one of our modality. Biological data was one of our modality. Imaging data and the biochemical data and the in vivo data, which we got from the animals studies. So this is the network, I would say, deep learning network. So if you could see on the left side, these are the red dots. We can say these are the input nodes. Each and every circle is called as a nodes, or you can say nodes, in the layman terminology, and these lines are the edges. So this every dot is an input node, and this here at the right side blue dots are the output nodes, and the orange color dots or orange color circles are called each line is called a layer, and all this network is called a deep neural network. So this is how neural network looks, but not exactly how it looks. So because here all nodes are connected with each other with the edges. Each and every node is connected with this red node is connected with all the red nodes are the connected with the orange circle. Let me get my, yeah. Yeah. So yeah, if you could see this red node is connected with this orange with if you could see this all red nodes are connected with this orange node. So this is a that's why we call it as a fully connected network or fully connected, uh, you can say fully connected network. So we have one, two, three, four, five input parameters, and this network is going to predict four output parameters. So you have to define how many outputs you want. Let's say if you just wanted to predict whether my molecule is toxic or non-toxic, then you can have only two nodes in the output layer. This is called output layer and this is called the input layer. So you can define only two layers in your output layer, two nodes in your output layer, and base these are the features. These nodes are input layer are we usually pass the features. Features are like, if we are dealing with only chemical data, then our features are like molecular descriptors. If we are dealing with the protein data, then we our features, in my case my features were uh, uh, fragments, or I would say fingerprints. Those were my features. So in my case I had a lot of features. And again, this layer definition, AI scientist, he will understand the data, he will analyze the data, and then he will define what kind of layers we should have or he should have. So we analyze for our data set for all these parameters. I cannot reveal, but for yes, in next slides I'll show you how we design network and what kind of networks we have used to build a model. Okay, so as you guys know, for the let's say for from the phase one studies, from the phase one clinical studies, we can get all this information, right? But for the phase two we cannot get. So we have to extrapolate all such kind of data for the longer term studies. So for the solution testing, that's why we have used only chemical data and the biological data. And again, we have passed this data for the testing, and we got the results. So I hope you must know about what the training set and the test set. If you don't, I'll be happy to explain. Let me explain you guys if you don't know about it. So let's say you have 100 samples, 100 data points, or 100 molecules. What exactly happens is you take all these 100 molecules, you calculate the fingerprints or descriptors for those 100 molecules, and then sorry, anybody has any question?
Sir, you can continue your talk.
Yeah. So let's say you have 100 molecules, and if you have calculated around 1,000 descriptors, so you have a matrix of 100 into 1,000, then you will divide your data set into 80 to 80-20 ratio or 70-30 ratio based on your data analysis, based on your experience, and completely based on you how you wanted to divide your data set. So if you are dividing your data set into 80-20 ratio, it means you are keeping 80% of your data for a training and rest of the 20% data for the testing. So this what exactly happens, so you use this 80% data for a training means algorithm, all these algorithms are actually machine learning, deep learning algorithms are the data savvy algorithms. It means we need data to train. We algorithm need data to understand. AI is what? AI needs the data, previous experiences, like a small kid. AI is like, you can say AI is like a small kid. The kind of data you feed in into AI, the kid will, or in a kid, the kid will understand in that way, or kid will know, okay, this is what happens. This is called spoon, this is called mobile, this is called bottle, this is called table, chair. The same happens with the AI. If you tell AI, okay, this image is something like a table and this is a table, AI will understand, yes, this is a table. If you give an image of a mobile or a spoon, if you tell him that yes, this is a mobile, this is a spoon, AI will understand, yes, this is mobile, this is spoon. So likewise, you have 100, as I said, you have 100 molecules. So AI understand, okay, out of this 100 molecules, few are the toxic molecules and few are the non-toxic molecules. This data we have to feed in, and then uh, the training happens on the 80% of the set. Means AI will understand, or this machine learning will understand, yes, the molecule, if the molecule looks like some, let's say something like this, then we can call it as a toxic. If the molecule looks something like this, then we can call it as a non-toxic. This is what AI understands from your input data. So this is exactly happening, and then rest of the 20% data, as I said, for a testing, we usually keep aside 20% data from the entire data set, and then once our model is built from on the training data, we pass this 80% data to predict, because this is the completely unknown data set for this model, right? Because we haven't passed this data set, we haven't used this data set for training, or and model doesn't know about this 20% data. So then we pass this 20% data through this model and will predict, let's say, in this 20%, we have 10% toxic and 10% non-toxic. So then we will check how this model is predicting for my rest of the 20% data. How many it is predicting this from toxic as a toxic and from non-toxic as a non-toxic. So there are several methods to I mean several mathematical formulas, I would say, to find out whether what's the accuracy. If you know about the sensitivity, specificity, recall value, F1 score, all these things we are using to find out the accuracy of our model or the predictability power of our model. So again come into the what we have done. But please do ask questions if you have any in the basic terminologies or anywhere in about the conceptual basis. So now, yes, and definitely I'm gonna keep lot of time for the question and answer after my session, because I really wanted to help you with all your problems. And also we can discuss if you are using AI or data science or any programming technologies in your studies, and if you are facing, please do come up with your problem as well after this session. And also I would like to discuss many points about how industry is using AI or data science or how they are automating all the process using programming in industry. So please do ask questions even after the session as well. So I'll keep a lot of time after the session. Now again, coming into the work, what we have done. So we have divided all our work into several phases: phase one, phase two, and phase three. So in phase one, we took only chemical information and the biological information, and always our labels will be toxicity endpoints, whether this molecule is toxic or non-toxic, what's the severity of this toxicity. So in phase two, we have used chemical and biological information, but along with that, biochemical information, biochemical parameters. So when if you can definitely ask me what are these biochemical parameters. So biochemical parameters are like, uh, urine analysis, blood sample analysis, and all other biochemical values for in the captured during that study. And in the phase three, we thought of using again the same chemical information, biological information, along with the toxicity endpoints from the short-term studies. We thought of using those data from a short-term study, from let's say phase one or a 1 to 3 months preclinical trials, along with the doses information we wanted to include into this. So I have hidden few steps over here because those might be a company rights or something. So but definitely I'll explain all things, how exactly AI is helping uh, drug discovery. So the final outcome is always toxicity. What toxicity, how, whether it's a toxic or non-toxic, whether this particular molecule or this study will fail in the next phase trial or not, and all. Also, this is an end point, final point. So the experiments, what kind of experiments we have done is like, data sources. We have used a lot of, as I said, using NLP, we got a lot of data from the public literatures and public databases, several open source databases, and also we got a lot of information from the very well-known databases like CIDR, FDA, PubChem, ToxRefDB. So once we have a data, we clean that data. We got, in some cases we got smiles, some cases we got chemical moieties, I mean the structure of a molecule in a 2D image. We convert you, if you know about, there are a lot of Python packages which can help you to convert your 2D image into smiles or a inchy key, or again you can convert your inchy key or a 2D inchy key or a smiles into your images. So if you know about smiles and Inchiki, very good. If you don't know, please go and check. These are the 1D representation of your any chemical moiety. So like in a protein, we call it as a FASTA sequence. We call it as a 1D representation, and you have a 3D structure of a molecule, right? Likewise, for the small molecules, for the drug molecules or of small molecules or chemical molecules, we call smiles as a 1D representation of a molecule. Then once we have all these molecules, once we clean these molecules, we calculated 2D and 3D descriptors and fingerprints of those molecules, which are around 21,000 2D and 3D descriptors. So if you know about descriptor, that is really good. But if you don't know, so let me give you a small definition of a descriptor. Descriptors are the molecular features. So 2D descriptors are like molecular features, like log p values, molecular weight. These are the molecular features. These features we call it as a 2D features or 2D descriptors. 3D descriptors or 3D features are like if you convert your molecule into three-dimensional space, or if you look your molecule into three-dimensional space, the chirality and conformations, all those angles, we call it as a 3D descriptor, 3D features. And another terminology is fingerprint. Fingerprints are something, let's say, substructure, identification of substructure, I would say directly. So let's say if you have a carboxyl group, then it will give 1. If you don't have carboxyl group in your molecule, it will assign 0. Likewise, if you have amino group in your molecule, then it will assign 1. If you don't have, then assign 0. If you have indole ring in your molecule, then it will assign 1. If you don't have indole ring, then assign 0. So, all these packages are available to find out, to calculate this descriptors. Just a minute, I'll have just water. Yeah. So so we have calculated these descriptors and fingerprints, which are of around 21,000 or 21,000 and a half. Using some packages, as I said, there are several Python packages. You can This is a Java based tool, Paddle. This is an RDKit. You can directly use your RDKit using your Python. You can call RDKit from a Python, and there is another software called Moderate, which will also can be called by Python or Java. So the once we so as I said, we have 21,000 molecules. So it means we had a matrix of around, let's say, X number of drugs and into 21,000, X into 21,000. Directly, you can say this is our matrix. I'm not revealing how many drugs we had in our study. So once we have this matrix, what the next step, what we have done is, missing values imputation, because for some molecules we may not get 3D features out of, let's say, if we have calculated around 5,000 3D features, so we may not have got 100 or 200 or 1,000 features for any particular molecule. So that's why sometimes we have to remove at least those features or those molecules based on the data availability in that matrix. Then we did dropping constant values column, removing duplicates columns, features transformation, then feature selection and feature scaling. So these are the first four steps are something like data analysis steps, I would say, which everyone has to do once they have all such kind of data. But once we have a clean data or a set of data which we have analyzed or processed, then we have to go for a scaling or the selection. So it depends on which methods we are using, so we can use either of the step. So let's say first we are selecting our features. So as I said, we have got 21,000 features. All these 21,000 features are actually not important for your model. So let's say if I'm working on a skin toxicity or skin studies, one of the most important features for me, when I talk features, these are either descriptors or the fingerprints. Okay, usually we call those information as a feature in the machine learning language. So in the I would say in the data science language, but for biology and chemistry we understand it as a descriptors and the fingerprint. So, as I said, we have calculated around 21,000 features. So, this 21,000 features may or may not be essential for my study or for my toxicity, right? But yes, as a being a pharmacy guy, being a person who knows who knows biology or chemistry, what I can directly say is yes, molecular weight and log P could be an essential feature, essential parameters for my study, right? So as you know, if we have a large molecular weight compounds or large molecular weight molecules, they cannot easily permeate through your skin layers. Correct. Maybe from dermis or subcutaneous layers, and along with that, if you have uh, and along with the log p values, log p, if you know, water and oil partition coefficient. So, so these parameters are actually essential for the skin. Along with that, a non-essential uh, is something like uh, maybe total polar surface area. We can say it could be a non-essential, but I'm not sure right away. But yes, so there are those features which are actually non-essential, we can remove those features. Or let's say all my molecules have a indole doesn't have indole ring, and this softwares or this package they have predicted yes 000 for all my molecules, then definitely this is a non-essential feature for me. Anyhow, my molecule doesn't have indole ring in all the molecule, so this is non-essential for me. At the same time, maybe all the toxic molecule may have pyridine ring, and non-toxic molecule may have let's say carboxyl group in somewhere in their moiety. So this feature is actually important for us because it is differentiating uh, my toxic and non-toxic molecules into whether if my molecule has an indole ring, then it is a toxic. If my molecule or a pyridine ring, it is a toxic. If my molecule have a carboxyl group, it's a non-toxic. So it is directly differentiating. So then in that case, this feature is actually important for us, right? So this feature selection is a step wherein we are selecting important features for our study. So out of this, we just gone forward with around 1,000 features or so. So and then once we have a feature, we have to do feature scaling. Feature scaling is something, we are assigning all the values into a some range for all the features. So as I said, we have done a lot of data analysis, feature analysis before directly going for building a machine learning model. We have to do sorry, we have to do a lot of data analysis, a lot of feature analysis, which features are important statistical way and some biological and chemical way as well. So we have plotted a lot of data. So this point is what it actually defines. It's a toxic or a non-toxic. All these points are only for a toxic or not. This each point is, you can say for this presentation, you can say each point is defining a drug. So we were just finding out, okay, what is this, whether it's a non-toxic because it is away from the clusters. So this is, if you have a bunch or everything segregated in one place, you call it as a cluster. So we were just analyzing all these molecules, whether this one is toxic or non-toxic, why it is coming out of the cluster, how we can bring this molecule into the cluster, if it is a toxic or if it is a part of this cluster. So we have done a lot of analysis, and we got few data points. Finally we have selected few data points over here for this study. Although the numbers are not accurate right now because we cannot reveal all this information. So Now the exact process comes in which I talk about. I have already given you the basics how exactly happens this model development process. So I'll just go through what exactly we have done for the chemical and biological data. So let's say we have a chemical data, those features, we have a biological data, protein features. We have using a chemical structure, we find out the toxicity. So what exactly? So let's say, as I said, we had a original data which we gathered from various sources, databases, literatures, and all of the sources. Then we curated that data using programming, using programming and all other stuffs. Then we have segregated. So yeah, as I said, we can divide our data into training and test set, but alongside, if you want more robust model, or if you really wanted to find out the more robustness of your model, you can also divide your data set into training set, test set, and the validation set, or you can say external evaluation set, whatever you can call it. But yes, you can divide your data into three different parts, wherein initially you directly keep aside your 20% of your data set into a validation set or the evaluation set, and then you use rest of your data for the further process. I would say, let's say 80% of data for the further steps, and then out of this 80%, you divide your data set in, let's say again, here let's say 60, and here again 20, because 20 already we have kept here for the validation set. So out of this 100 molecule, let's say I have a 20 molecules here, and then 60 molecules here as a training, and 60 molecule for the 20 molecules for the testing. So again, as I said, you have a 60 molecules in your training. You can divide your data set into various different parts. So uh, like you can use lot of leave one out method, wire randomization method, combi QSR method. Definitely you can Google out these terminologies. I'm not gonna go detail in these are very basic terminologies, but yes, using these models, you can use machine learning algorithms to develop these models, and these models will help you to build predict. I would say models called as a prediction models. And then once you have your model, you usually pass your training data itself to predict the robustness, or I would say the predictability of your model. So that is called training accuracy or training validation parameters. And once you have very good training accuracy, we usually call, if you have more than 90 or 95% of accuracy, we usually call it as a very good model for the training data only. And once you pass your test data into your model, this is an unknown set, right, for your model, for this model, for this model. So this is an unknown set. So if this model could able to predict this unknown set, let's say out of this 20, if it could able to predict 16 molecules at least, then at least you can say your model is at least 80% have an 80% accuracy. Along the same side, if you pass your validation set over here, and if it also could able to predict it around 16 molecules, then you can say, yes, my model is very robust. It could predict unseen data with an 80% of accuracy. Then definitely you can go ahead with this model, and you can predict unknown molecules. You Very unknown molecules. Let's say if somebody is doing lab experimentation, or somebody is doing any lab studies, he can directly pass on these molecules over here. So so this is how model development, training set, test set, validation set happens in a AI space for a chemistry and a drug discovery. Okay. Now what we have done. So as I said, we tried with a multimodality approach. So what we have done is something. Initially, as I said, we calculated the chemical descriptors, and we built this model in this steps in this procedure. We also converted our chemical molecules into a image format. 2D images. These are completely 2D images. If you know self-driving cars, Tesla cars, or automated driving systems, ADAS or something, so you know, you guys might have heard about car gets stopped if it finds any object in a road. This self-driving cars, right? So car can get diversions if it finds any object, or he can pass it with he can pass by side if the car finds any object. So how this exactly happens? This happens with the deep learning. I would say deep learning. This entire field is called the computer vision. So computer vision is exactly what exactly it is. It understands the image data. We have to feed the image data to the system, or you can say algorithm, and algorithm understand based on these images. Let's say I can give thousands of images of a different cats to the algorithm, and I can say yes, these all these animals which are look like a cat and which are actually a different form of a cats are actually cats. Machine understands, okay, cat look likes in those thousand forms. And if I give another 1,000 or 2,000 images of a dog, different spaces of a dog or different angles of a dog, and in that case machine understand, okay, this is how dog look likes. And if you ask machine sometime, if you give another image, let's say you just click one image of your cat or a dog and you feed it into machine, your machine will tell you, okay, this is a dog or a cat. So this is how the driving system works. So if any object comes in, you know, these cars have these cars have camera, and at the back end they capture all these images. These images goes to the system, and system predicts, okay, this is cat or any animal or object or anything. The system predicts. So this field, the entire field is called the. If you are dealing with the images data, then the field is called the computer vision. If you are dealing with the textual data, the field is called natural language processing, or NLP. And uh, so all these fields can mostly in the computer vision, and mostly in for the NLP, people are using deep learning algorithms, because these deep learning algorithms require a lot of data, and they are really found to be predictive, and they are giving very good results for those image data and textual data. Okay, now coming into the work again. So we converted our molecules into images format, and we have applied this model. This model, you can say, is a deep learning model for the computer vision, you can say. And again, we got, as I said, some textual information. These are the smiles format. So these are the 1D representation of a molecule. We converted all the molecules into a smiles format. Along with that, we have used the protein sequences for our study. So this protein sequence information plus smiles, we have combined this textual format, and we built a NLP model, our deep learning model for the in the space of NLP. We again, to build an NLP model, you have to do a lot of data processing steps. You have to clean your data. Again, you have to identify molecules. You have to standardize all your parameters. Then you have to uh, so for the molecules, the features are like words or the sentences, right? Sorry, for NLP, the features are like words and the sentences. So in NLP, we use mostly uh, embedding algorithms to derive the features from those sentences from the text. You can definitely go into what are those embedding algorithms, how and there are several basic methods, like, you know, count factorizer, TF-IDF factorizer, if you have heard about, to convert into features. Then we are using those features over here in the case of NLP. And then finally, we are predicting what is an output using NLP. So you could see, right? We have molecular features, numerical data. We had images data, and also we had some textual data. So you can call it as a multi-modality approach. So wherein we are not only dependent on the numerical data, sorry, but also we have used textual data, and we have used the images data for our study. So if you are using NLP, if you are using computer vision, all, and if you are using normal machine learning algorithm all together, then you can call that study as a multi-modality study or multi-modality approach. So I would say there are very less papers who talks about multimodality right now in PubMed or something. We are very fortunate to work. We are really fortunate to work on such modality, and we got very good results actually. I'll show you in the next slides. So as I said, for the, let's say for the binary classification, there are several machine learning algorithms, but we use XGBoost. Although we have used a lot of, but we got very good results with the XGBoost algorithm. Again, you have to you in those algorithms, like also you have to set up a lot of parameters. You have to set out your parameters, you have to understand your data. Based on your data, you have to define your parameters. And also, once you define your parameters, if it is not giving you the very best result, again you have to optimize your parameters. Again, to optimize parameters, there are again few different algorithms. So this is very complex process, guys. So you can use other different algorithms to optimize your parameters, or you can define your own parameters for the building the model. It's completely based on you, how you do it. And again, as I said, you can divide your data into training and test and validation set. But yes, how to use this training data for your training? There are several methods, like cross validation method, leave one out method. So we tried all those methods which are available to use different combination of our data points in our training set. And then metric selection, as I said, accuracy. But in deep learning, you can say you usually calculate the log loss, or and also you can find out your values in logistic regression. You calculate you find out your values for the softmax. I mean, although logistic regression or the softmax gives us 0 and 1, but still you can find out the probability, how much is the probabilities, whether it's 0 0.5, 0 0.7, or 0 0.3 or 0 0.1. So we did all this analysis. And for image data, we have used ResNet. CNN is So we for this data we have used ResNet. ResNet is a, you can say, already pre-built network. ResNet is already pre-built network wherein you have to map your data, and again you have to tune your lot of parameters. So I don't know if you know about encoder, decoder, these terminologies. You can say these are part of uh, uh, computer vision algorithms. You can say these are terminologies for the AI. So what we have used is, so let's say this values, this blue color, these are the convolutional layers. Okay, a convolutional neural network. If this network, you can call it as a CNN, convolutional neural network, and this layer you can call it as a convolutional layer. Then we have a ReLU. ReLU is the activation function. If you know about activation function, that is good, but let me give you brief about activation function, what exactly it is. As I said, this layer is something like this one. This layer, you can say something like this. Okay, a network, all these layers, but not exactly this. This is just for an example. And whatever, let's say, whatever values you are getting over here, we have added activation function over here. Okay, so let's say if you are getting a probability of 0.5, then this 0.5 goes into the activation function. Here, as I said, we have used softmax. Softmax is an activation function. So here we have used the ReLU as an activation function. So softmax is something like S shape. This point is 0.5. If it is below than this, then it is below 0.5. It is more, it assigns always zero value. If it is if any my probability is less than 0.5. If my probability is more than 0.5, it assigns 1. So this is softmax function. What tan h ReLU, leaky ReLU does is, let's say you have x-axis and y-axis. So you are getting some values over here. If any positive value, you capture all that positive value. In softmax you are using uh, you use those values, you convert all those values into zero in the range of 0 and 1. But in Relu or Tanish, you take all those positive value as is, and negative value always you put 0 in case of only Relu. On Licky Relu, you can convert your values into negatives as well. So these are the functions of activation. These are the functioning of the activation functions. And then you can input one another layer for the patch normalization. So you can say this is a single network for our purpose. So likewise we had a lot of those networks, so you can understand how complex this model is. So here in each layer, in each node, we have several different normalization functions, several different weights, constants, everything. So this is how the complex networks looks like. And then, so if you aware about this, we cannot build such models in our normal computer having let's say 16 or 32 GB of RAM with a lot of size. It doesn't. It always requires a GPU and a lot of computational power. So this calculation usually takes a week of time. Only model development process may take week of time, or maybe 10 days or 15 days, based on your again computational power. So nowadays you must have heard about the cloud technologies like Amazon, AWS, Amazon Cloud, or Azure, which is offered by Microsoft, and all other companies have came forward which are offering cloud technologies wherein you don't have to purchase your own big servers. You can I would say rent it out, those Amazon clouds. You just buy for some days, and you pay for the pay cost for some days only, and then you turn off your system. Then you don't have to pay. So that is Amazon service. Azure, these services are like pay per use service. uh, if you are using those servers or those machines for let's say 1 month, then you have to pay only for a 1 month time period. So this is how we have built a model. So let me go somewhat little bit fast now. So we got as I said, we developed several models. So with XGBoost we got 53% of accuracy, with RFC we got 63, and with Tabnet we got 57% of accuracy. And as I said, we developed images data with the ResNet model. We got 63% of accuracy. And if you combine this all these accuracies, uh, this is called ensemble model. If you combine multiple models into one, called as a ensemble model. So then we again search for another algorithm called graph neural network, which I said, for the node classification and age prediction. Graph is something look like this. A molecule can be represented in this form, in a graph format. Each node will define the atom in a molecule, and each this age defines a bond in a molecule, right? So along with that, we have used same for the protein sequence, and we this you can say a proteins graph. Each this is an amino acid, or atom in amino acid, based on the what kind of graph you are building. Then we use this GNN technology, and this is again graph neural network is again an algorithm. We use this algorithm to predict whether it's a toxic or non-toxic, and then everything actually requires. That's why in the initial of this session I asked all of you that if you know Python. Mostly I do Python. I'm working on Python since 2011, during my from my master's time. So still you can use a lot of data science packages in R as well. But yes, mostly the common language for AI. If you really want to do work in AI, if you really want to contribute to the AI, or you want to incorporate AI in your daily research work, then you have I should say you have to learn Python. And learning a Python is not a big challenge. I have found a lot of people who learns Python in 1 month or less than a month. So even those are from the pharmacy, those are the pharmacy guy. right? So if you don't have a degree in computer science or degree in engineering, still you can learn Python very easily. Python is a very good language, useful for the data science and can be learned easily. So this is what we have done, a lot of things up with the graph neural network, and then this is how exactly calculations happens in a graph neural network. And as I said, by combining previous models, we got only 71% of accuracy. But when we add our graph neural network model into this ensemble with some weights, the accuracy has got drastically improved to 91%. Initially, we had only 71%. Now, adding this model, we got 91%. So this study concludes that if we add multiple modalities in our model, we can get very good accuracy compared to any single modality, any single mode. So in a phase two, what we, as I said, we use in silico data, which we have developed here, the chemical and biological data, we usually call it as in silico data. And then we have used in vivo data and imaging data. We work on a lot of pathological images of the from those samples, animal samples like skin rashes. We got some samples, then we developed some same such kind of models. This such kind of networks we have developed using images. And then in a phase three, what we can use animal data, then chemical information, time duration, dose information. So if you have, let's say, this is an x-axis and y-axis. Y-axis we have a dose, x-axis we have a duration in a month. So we can get only from a 1 month or a 3 month study data, right? But LSTM is an algorithm, you can say is a part of NLP gain. So you can extrapolate this data for the 9 months or for the 10 months, dose data with the duration. What dose we required with respect to data from the short-term data. This you can extrapolate with LSTM. LSTM can helpful for this information as well. So this information uh, also LSTM, if you have heard about LSTM, is many people have used for the stock market prediction also. Let's say if you have a data for last year, you can predict what could be the stock price for that stock price for the company next month or next days or next in next second as well. Machine learning can help you to predict the stock price using LSTM, and there are even more stronger algorithm than LSTM as of now in the market. And also we have done a lot of scaffold extraction, substructure extraction, and then finally we predict the long-term effect of those studies from the shortest term studies. So and now I'll quickly wrap it up. So this is the software tool. I just wanted to show you what we have done so far in this field. So this is a tool which I have developed in 2013 with Persistent Systems Limited uh, as a part of my master's work. So here, let's say if you give me molecules, these are the SMILES molecule. If you give me this molecule, my tool will find out whether this molecule is a sensitizer or non-sensitizer, what mechanism, by which mechanism it is acting as a sensitizer, what's the similarity of this molecule with sensitizer or non-sensitizer, and these are some domain-specific values for this molecule. And also, let's say this molecule, first molecule is the structure for this first molecule is this one. So with the programming or with the AI, we could able to find out which part of this molecule or which substructure of this molecule is actually causing the skin sensitization. So we have highlighted it with the cyan color. So this software was really helpful for many people, those those who were working on the skins, and we have published the paper in PLOS One journal as well during that time. Also, during my PhD time, I have developed a software tool called CameSuit. if somebody doesn't have a knowledge of machine learning, if somebody doesn't know Python programming, still he can develop a model. This is a panel wherein home tab we have a calculations tab, we have a processing development, predict analysis, and about us tab. So you just have to import your molecules. You have to select what kind of molecular features you have to calculate for your molecules, like RDP, PyDPI, descriptor, fingerprint, paddle, and then you just click on a run. You will get all those features of your molecule. Each row defines a molecule, or you can say protein, and each column defines the features or a descriptors. So and then at the back end, I wrote a lot of machine learning algorithms using Python. You don't have to write any program, any algorithm. You just have to do click. You just have to select the parameters. You have to enter input the parameter, and you have to click the button. That's it, nothing else. You A person don't have a knowledge of chemistry or a biology, still he can build a machine learning model in a 5 or 10 minutes. That's it, nothing else. We If you start programming, if you write your programming, you may take several days, but using this software you can build your model in only span of 5 to 10 minutes, even not more than that. If you have a good computer, means around 8 GB of RAM or 16 GB of RAM, and if you have a data points maybe somewhere around My screen is visible. Can anybody
Yes, sir.
confirm?
Yes, sir.
PPT
Is
is
it?
visible, right?
Yes, sir. Visible.
Perfect. So another database which we have developed. This is an interface. You can go through this link. This link will work from the evening. Right now it's not working, but yes, we have developed a database which can give you all this information, like adverse drug reaction information, wherein you just enter a CAS number or a drug name over here. You will get all the information about the drug, like you'll get all information drug, like drug name, pubchem ID, smiles, inchi keys, therapeutic categories, and all their drug interactions. So let's say this carbamazepine is interacting with allopurinol. It causes this particular toxicity. It causes this toxicity based on this interaction type, and by which mechanism it is causing this toxicity, and in which reference, in which paper this information is given. All this kind of information is available in this database. Along with that, we have a drug and protein interaction information, drug and food interaction information, drug metabolites, and ADR. So we have around this information for more than, I think, 2000, more than 2000 or 2300 drugs information in this database, and a lot of protein information as well in this database. Drug interaction, drug food, drug protein, and drug toxic metabolites, and their adverse drug reaction. And all this information is being gathered or collected from the very well known validated resources. Using, again, we have to gather this such a huge lot of information, we cannot do manually. If we go with the manually, we cannot do in a short period of time. So that's why here NLP can help us to automate this process. And NLP helped me a lot. Natural language processing helped me a lot to find out these targets, to find out these drugs, their interactions, their mechanism, everything from those papers. And so this game suit was part of my PhD. Another was part of my PhD. I have developed another software, the programs which I have written for natural language processing to prioritize those articles, to find out those information from those articles. I have developed a tool called eCider. If you just need to type any keyword, let's say cancer or something, you will get how many records are there. You can, if you type cancer in a PubMed, you'll get millions of articles. But if you type cancer in our tool, you'll get millions of particles, but this tool will help you to find out which article is the best one to read, because you cannot read millions of particles at one go. But this tool will tell you, okay, this is the article which you have to read first, because it has a more importance based on your keywords, or more important. Again, it calculates at the back end we have lot of sorry, at the back end we have lot of uh, 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 algorithm, or we have lot of calculations going on which defines, okay, this particular article is important or not. Again, you can get articles from the abstract or full text, or based on some time duration, you can get a lot of information. So yeah, thank you for your time and your patience for this work. Now I'll open this session for the question and answer. I'll be really happy to answer all your questions. uh, if you have anything, how you can use your AI machine learning in your studies, or what you can do with AI. So I'll be happy to answer, or what exactly industry is doing in the field of AI for the biology and chemistry. Yeah, I'll be happy to answer all your questions. Thank you, everyone.
Okay, sir. If anyone have queries, please raise your hand or post it on chat box. Okay. I would like to thank our chief guest, Dr. Sujit Tangarpalli, who honored this session with his inspirational thoughts. Now it's time to present certificate to our guest who filled our brains with his knowledge. Share our.
Thank you very much for the certificate, and thank you very much for this opportunity to present my thoughts in front of good people over here. Thank you, guys. Thank you, everyone. Thanks for
Thank
the
you. Thank
man.
you, Dr. Sujit Gadu, for the valuable lecture.
Thank you. Thank you very much. Have a great day to all.
Okay, thank you, Anandal. Dati Gadu, please post the feedback link. The participants are asking the feedback link.
If anybody doesn't have questions, so shall I leave the session or should I stay here?
Yes, sir. The session is closed, sir.
Thank you very much. Please share feedback with me as well.
Okay, sure, sir. Thank you, sir.
Thank you.
Thank
Bye.
you.
The assessment and feedback link is shared in chat box.